Hi, everyone. Thank you so much for joining us today. My name is Bernat Navarro. I'm a senior science policy analyst at Friends of Cancer Research. We are really glad to have you here. Before we get started, just a quick reminder that you can submit questions anytime using the Q&A box. We'll try to get to as many as we can. The session is also being recorded and will be shared afterwards on YouTube. We have a fantastic panel joining us today. And before we hear from them, I want to take a few minutes to provide some additional background on external control arms. Randomized controlled clinical trials are considered the gold standard for understanding whether a treatment works, largely because they can reduce bias. Patients are randomly assigned, which allows for more reliable comparisons. They also give us a clear way to compare new treatments to standard of care, generating the kind of evidence that informs regulatory decisions and clinical use. Over time, this is how standards of care are established and updated. But even with all of these strengths, randomized controlled trials aren't always feasible. There may not be enough patients to support randomization, especially in rare cancers. In some cases, there's high unmet need, and assigning patients to a control group may not be appropriate. In small or limited data, the evidence can be difficult to interpret. And there's also the reality that many standards of care remain unchanged for many years, limiting what comparisons are possible. So while randomized clinical trials remain essential, they don't always work in every situation. So how do we generate reliable evidence when randomized controlled trials are impossible? That's exactly where new approaches come in. One potential approach is something called an external control arm, or ECA. ECAs use existing patient data like electronic health records or prior clinical trials to create a comparison group instead of enrolling patients into a traditional control arm. These can be especially valuable in rare cancers or situations where enrollment is limited. ECA allow us to learn from real-world patient experiences, capture how treatments perform in routine care, and potentially generate evidence faster. But there are important challenges. Real-world data can be incomplete. Patients in these data sets may differ from clinical trial populations, and results may vary depending on how the data is analyzed. So while ECAs are promising, there's still uncertainty about when and how they can be used reliably, especially for regulatory decisions. So to make this a little bit more concrete, let me walk you through how an external control arm could be used in practice. We start with existing patient data. This again could be electronic health records or data from prior studies. And one important consideration is that patients in these data sets don't always look exactly like those from rolling clinical trials. They may have different characteristics, maybe treated in different ways, or some information may just not be captured in the same level of detail. So a key step is identifying patients who are as similar as possible to the trial population. That group can become then an external control arm, patients who will have received the standard care. In parallel, patients enroll in the clinical trial and receive a new treatment. Then we're able to compare outcomes between the external control arm and the treatment arm. So this approach could allow us to build a comparison group using existing data, but that again depends on how well we can account for those differences and ensure that the comparison is meaningful. That is where our ECA pilot project comes in. And at the core of this effort was a simple question, or complicated question. How can we reliably use external data to support the evaluation of new treatments, especially when randomized trials are infeasible or appropriate? To do that, we brought together a multi-stakeholder collaboration to test these in a structured way. We developed a shared statistical analysis plan so the data partners that were approaching this problem could do it consistently. Then each of data partners independently constructed an external control arm using their own external data sets. This allowed us to not look just at whether that is possible, but how consistent the results are, as well as where differences started to emerge. This goal was to generate practical evidence-based insights into what it could take to use these approaches reliably. This work, again, included 9 different partners, each contributing patient data from external sources. Our target trial aligned population was the control arm of the RESOLVE study. It is a completely randomized controlled trial in patients with metastatic pancreatic ductal adenocarcinoma. Again, this is a high unmet need case, as well as the standard of care hasn't changed in quite a long time. Patients received gemcitabine as well as nab-paclitaxel, and that will also be the adult population for our external data cohort. So this slide is going to walk through how the pilot worked. Again, each group started with their external data, like real-world data sets or prior clinical trials. They applied a very similar eligibility criteria as the clinical trial, the RESOLVE trial, to identify comparable patients. And they constructed their own version of an external control arm. Finally, we compared these results to the actual controlled trial from the RESOLVE randomized clinical trial. And these allowed us to assess whether ECAs are reproducible, how much the results vary across data sources, and what that means for real-world use. Here is an example of 4 different ACAs that were constructed. In black, you will see the survival curve for the control arm of the RESOLVE trial, and in blue, different external control arms that were generated. First of all, what we found is encouraging, but also highlights important complexities. First, ECAs can be constructed across different data sources using a shared statistical analysis plan. Patient selection really is going to depend on the data availability of those sources, and what is captured in those data sets matters a lot. The results that we see vary depending on the data as well as some of the methods used. And finally, transparency in all of our approaches is critical to ensure that these results are reliable. Understanding how data was selected, analyzed, and interpreted is essential to building trust in these results. So ECAs definitely show promise, but careful implementation and clear standards are key. So with that, I want to pass it to our fantastic panel today. We have Cassidy Moravec, the Senior Director of Strategic Collaborations and Patient Advocacy at the Pancreatic Cancer Action Network, Elizabeth Garrett-Meyer, Vice President of the Center for Research and Analytics at the American Society of Clinical Oncology, Ashita Vatavia, Global Head of Study Innovation at Johnson & Johnson Innovative Medicine, and Eva May, Friends Advisory Advocate. Thank you all again for joining us today.
Thank you, Bernat, and thank you for the invitation to moderate this wonderful panel of experts. Really looking forward to the conversation today. So as a reminder, as Bernat mentioned, feel free to drop your questions in the Q&A, and we will get to those at the end of the panel. So let's jump in. So the first question I have for our panelists. So in the ECA pilot project, the working group did select metastatic pancreatic ductal adenocarcinoma as the case study. Can you speak to why the setting was chosen and what it tells us about where ECAs are most appropriate today, and then what gaps they might help to fill in the future? So Liz, I'll start with you.
Great. Thank you. Thank you, Cassidy. So metastatic pancreatic cancer reflects a setting with high unmet need and real challenges for traditional randomized trials, particularly around the control arm recruitment. So using it as a case study really helped ground the pilot in a context where ECAs may be most impactful, such as rare cancers, or settings where generating comparative evidence is difficult. And it opens the broader discussion of where ECAs can help fill evidence gaps today. And from a measurement perspective, using a setting where overall survival is the endpoint alleviated the issue of potential variability that we could see in other efficacy endpoints. So for example, outside of clinical trials, there is subjectivity to declaring that a patient's cancer has progressed or has responded to therapy. Survival times are objective, so that was another reason that this setting, using overall survival as the endpoint, made sense for this project.
Wonderful. Thanks, Liz. And Ashita, do you have thoughts on this as well?
Yeah, first of all, thank you, friends, for the invitation to participate in this webinar. Thank you, Cassidy, for moderating, and I'm feeling pretty delighted to be on a panel with so many smart people. I agree with everything Liz just said, and this is such a great example of a use case where we can put forward ECAs to really advance our understanding. I think one of the things that might be worth also pointing out is, aside from this kind of use case where you're in a rare disease with, standard of care, there are other instances as well where ECAs can be impactful. I spent a lot of time thinking about multiple myeloma, and there's been just a burst of activity and innovation in that space. And we've got this sort of wonderful but terrible problem of our standard of care keeps rapidly changing to better and better treatments. Patients live longer and longer. Excellent. However, there's still work to be done. And by the time we decide on a control arm or a randomized trial, by the time the trial reads out, that's no longer standard of care. And so for those sorts of scenarios too, this can be impactful using real world evidence in ECAs, because then you have a way of comparing the intervention arm to the contemporaneous standard of care at time of trial readout, which is exciting. But, on the flip side, we also have more challenges without cleaner endpoints the way you have in prostate, pancreatic with like OS, where yes, we do OS and myeloma, but the IMWG and other criteria, there's new other challenges to consider.
Wonderful. Thank you for those perspectives. So the next question building off of that. So ECAs are already being used in some settings, but not yet consistently incorporated into the regulatory decision making. So what are the key challenges that are limiting their broader use? And how did the ECA pilot project actually help us illustrate these challenges in practice? So Ashita, I will go to you first for this question.
Thank you. Yeah, I think one of the really interesting things that was surfaced so nicely in this project was that when you use different sources of real world data, not all real world data for the same patient population is created equal. There's different factors that may be missing in data set A versus B versus C versus D. And how do you reconcile that can be a pretty interesting thing to think about statistically with a smart group of people. There's a variety of different solves, and that's really where some of the art comes into the science, which is good to see. I think another area, and we'll probably touch upon this later too, is you still have those consistent challenges, even with a clean endpoint like OS, of what is your surveillance bias, what is your misclassification bias, less so in that context, but in some other context, these are considerations that we have. But I would love to pass to Ava to add on.
Hi, I'm really happy to be here today, and shout out and hello to all the advocates that are in attendance. Constructing a credible ECA is really complex, particularly when working across data sets with different levels of completeness and consistency. This pilot really helps make this more tangible and shows how variability in data sources and the robustness of baseline information directly affect data viability. It also reinforces the potential that credible ECAs have to make clinical trial participation much more attractive to patients, especially as trials struggle to face so many challenges with control arm recruitment.
Wonderful. Thank you for those perspectives. So I'd like to dig in a little bit more on those challenges. So how is the field working to address these challenges and move toward more reliable use of ECAs in oncology drug development? And then to build on that, what role can patient advocates play in helping move this forward, including strengthening the data sources and ensuring the right baseline data are captured? So Liz, I'll start with you, and then we'll go to Ava for the patient perspective.
Great, thank you. Some of you who may know me, I'm a statistician, so I focus a lot on the data, a lot on issues of completeness and bias. So one of the things that I know that many of us are working towards is increasing the effort across the field to improve ECAs. The way that they're constructed and evaluated really requires having better alignment on methodologic approaches and a very intentional use of the real world data. So we have to understand really when ECAs are truly fit for purpose. And that's going to require us to not just have more data, but we need higher quality data, more complete data. And this particular pilot, I think, really highlighted that, and we can use this to help emphasize that in the field. So just to sort of sum that up, we learned that we really do need complete data because that's where we saw the best alignment, and common definitions for the key variables that we need to create the ECAs.
The pilot also made clear that having strong baseline data is critical. Without it, it's really, really difficult and sometimes impossible to build a credible external control arm. Patient advocates and advocacy organizations can help play an important role here by helping identify where the data gaps exist supporting efforts to improve data collection, and contributing to the development of data sources, especially in rare diseases where existing data may be limited but highly, highly valuable.
Wonderful. Those are definitely extremely key points. So thank you, Ava. So doubling down on those external data sources. So as Liz, as you mentioned, the ECAs do rely on those external data sources, including real world data. We all know that that can be a little bit messy sometimes. So drawing on the ECA pilot project, which used retrospectively collected data across multiple sources using a common statistical analysis plan, what have we learned about the importance of that baseline data, the differences across those data sets, and how retrospective as well as prospective approaches compare in constructing reliable ECAs?
Yeah, so I think the pilot really highlighted that while retrospective real world data can be a powerful resource, using the different data sources that we had, it does introduce significant variability in how the data are captured, especially at baseline. And the lack of baseline information appeared to contribute to poor agreement with the trial's control arm. And we went through the results pretty quickly, but some of you may notice the difference in the curves. Some of them were right on top of each other, and then for others, there was a more notable difference. So differences in the completeness, the definitions, as I mentioned before, and the structure will directly affect whether a credible ECA can be constructed. And I think that we also were able to underscore the trade-offs between the retrospective approaches that were used, which some may be more feasible, but also we had some heterogeneity. And the prospective approaches that we may be able to take, they could allow for more consistent and fit for purpose data collection in projects that we could look at moving forward, where we have prospective data collection versus retrospective data.
That's great. Thanks, Liz. So, Ava, from a patient perspective, what do ECAs mean in practice, particularly in the terms of control arm assignment? We know that's a big topic for patients when they're looking into clinical trials, as well as what does that mean for clinical trial participation, as well as access to new therapies and new treatments for patients?
So one of the most important implications is around the experience of enrolling in a clinical trial. Patients always feel like they win by getting into a trial, but they lose if they're then assigned to the non-interventional control arm. And what's really exciting about ECAs is that they can actually replace or at least augment traditional control arms, which can be particularly useful in trials where enrolling patients into control arms is challenging, which we're seeing more and more.
Wonderful. Thanks, Ava. And Nishita, can you add to that?
Yeah, absolutely. I think there's something really interesting about this idea of winning when you get onto a trial and enrolled, and then losing when you get put on the control arm. And I think it's a tough concept as like a scientist or a statistician to wrap your heads around, but from a very human standpoint, it makes complete sense. And I think, as there is more opportunity for ECAs, it doesn't necessarily mean that we walk away from randomized clinical trials. And I think that, or randomized control trials, that's what Bernat was saying at the beginning. I think it's just an opportunity to maybe move towards hybrid arms in certain situations, or really think about in just the natural state of care in the world and, standard of care and what patients receive, there is still so much value and insight that can be drawn from that, and being able to leverage that data, have smaller control arms, potentially give you opportunity to accelerate enrollment. I'm not sure that how widely known it is. It was news to me when I started working in research, that the longest part of a trial often is that enrollment phase. It's really hard to drive enrollment forward. And if you can leverage existing data to do that, that means that you get there faster, you get your insights faster, you get treatments could be approved faster, available to a broader swatch of patients faster. There's a lot of reasons why this makes a lot of sense, but then, having the hubris to understand, and I'll put my doctor hat on for a second. One of the things as I was listening to my fellow panelists and reflecting on is, you know, sometimes the translation of what it means for missing data in the real world to real examples can be a little bit trickier to wrap your head around. But a good example of this is that, as a physician, my note doesn't always look like every other physician's note. So I'm capturing it for a totally different purpose than research. I'm writing it as a communication document of what I think is salient for that patient's care moving forward. It's not necessarily with the intention of informing a trial later. Trials, by definition, are collecting a copious amount of information to get a different answer, and not every feature of what they're asking is captured in the real world data. And that's the origin story of a lot of the missingness that we're talking about here. It's that stylistic difference in the patient, physician, patient, healthcare provider interaction. And so having awareness of that is important. And I think, it was just something I wanted to insert into our conversation here.
Can I riff off of that as well? And I mean, I've thought about this a lot as a statistician, because in the clinical trials world, when we think about missing data, we think about a question on a CRF that does not have an answer filled in. But when we talk about completeness or lack of complete data, I try not to call it missing data, because there's nothing really, as Ashita just said, there's nothing really missing about it. An individual care provider did not put something in the chart because maybe they didn't feel that it was necessarily relevant or something they didn't need to follow up on. So, so trying to create these complete data sets is very challenging, because we don't necessarily even know what completeness means before we are starting to look at the data sort of in a retrospective way.
Thank you, Liz. Those are very excellent points. Rashida, did you have something else to say?
I did. I wanted to just tack on, Friends has also done a lot of really exciting work with pragmatic trial design, where you're really paring down what you need for a trial and for analysis. And I think there is some measure of synergy across like a slimmed down, like not nice to have, really keep it at need to have, and then having a little bit more theoretical opportunity for ECAs if you're comparing something like that. So there is that kind of piece of it that I sometimes think about, because that's what I do in my free time. But I think it's an interesting thing to position out there for a broader audience.
Absolutely. And I'm sure we'll get some questions about that in the Q&A. So we'll come back to that a little bit more later. Wanted to kind of zoom out and touch on, Ashita, some of your comments about those notes between patients and providers, and kind of what gets into our real world data. So how are patient data actually used in ECAs, and what should patients understand about privacy, consent, what's in those notes, and how their data contribute to research? So Ashita, I'll start with you.
Yeah, it's a really good question, and it's a really important one, because I can see people getting anxious. I'd be anxious too about, hey, what are you doing with this data that's between me and my provider, right? But I think when we develop these data sets, these real world data sets that we're using, these fit for purpose data sets, they're essentially a pre-specified level of fields, and we're collecting anonymized data from these charts. So privacy is front of mind, I think, for all of our data partners, our researchers, everybody that's working with this data, particularly regulators, if they're considering it. So that's something that, deserves a lot of time to carefully answer and address questions. And Ava, I'd love to hear your thoughts as well against this one.
I could talk a long time on this, but from a patient perspective, there's often a great deal of uncertainty about how data are being used in these contexts. Helping patients understand that their data contribute to research in meaningful ways, and that they're still part of the process even when their data are de-identified and are going to stay de-identified, can help build trust and make this work more accessible and relatable.
Wonderful. I think that's a key patient perspective. Thank you, Ava. So based on the ECA pilot project. We heard a bit from Bernat. We've heard from you all the different perspectives. So how has your perspective evolved since the beginning of this project to where we are now? So are you more optimistic about the role of ECAs today than when the work began? And if so, why? If not, why? So Liz, I'll start with you.
Yeah, thanks. This is a great question. So I think I already divulged that I'm a statistician, and one of the things for those of you who have worked with statisticians before, we all often tend to be natural skeptics or Debbie Downers. We come to the table with uh, maybe not a lot of optimism for some of the things that could go wrong. And we're thinking about biases and all sorts of challenges. So I was really not that optimistic at the outset, but of course, very interested in being a part of this and seeing how it played out, and also inserting my skepticism early on, hoping that that might be able to help us develop a plan that would address some of those potential problems and pitfalls along the way. But I have to say the results here have been extremely encouraging to me. So for some of the data sources, we saw that the ECA outcome curves were almost indistinguishable from the trial control arms. And it turned out that those that matched most closely tended to be those from data sources that had the most complete baseline data that allowed us to implement appropriate matching. And we ended up also having this unintended sensitivity analysis, really because there were some data sources that had more limited data. And what we saw was that they didn't actually match up as well. So if we had less data and less ability to do what we might call more stringent matching, we did see that those curves did not lie quite as closely on top of the control arm that we were trying to match. So if all the data sources had similar completeness, we would not have had a chance to see how that might have played out. So that was actually, we talked a lot in our planning meetings about doing different sensitivity analyses. And, in the interest of time, we couldn't do all the things we wanted to do, but it just turned out that because of the heterogeneity of the data sources, we were able to really look at that hypothesis, which I think gave me, it really encourages me for this approach for the future.
That's great that you are now optimistic. That's amazing from a skeptic. So Ishita, would love to hear your take on the applicability of these insights across oncology indications.
Yeah, I think I agree with everything Liz said. I would only add that I am more optimistic as well. And even though the work was in pancreatic, it was really easy to see how the lessons and what we were surfacing had applicability more broadly across oncology.
Wonderful. And Ava, would love your patient perspective.
So during the event, the panelists talked about the difficulties of matching real-world patient data to clinical trial data in terms of the patient eligibility criteria typically seen in a clinical trial. And one panelist said that in the future, we should not only strive to fit the real-world data to optimize the trial, but to optimize the trial to match the real-world data, i.e. create a pragmatic trial so we can enroll a more general population and make the balancing between trial patients and patients in external control arms much easier. Cassidy's nodding. This kind of was the big takeaway for a lot of the advocates that attended. I'm optimistic that the evolution of clinical research to include real-world data will result in the acceleration of access to new and better treatments that work for a broader array of patients in their real-world settings.
Wonderful. Thank you, Ava. I completely agree with all of the panels. And I have one more question before we get to our Q&A. I see some questions coming in. So looking ahead, what needs to change across the data, the methodology, or policy to make ECAs more reliable, more widely used as a tool in regulatory decision making? And how might ECAs also be used beyond approvals to better understand how treatment affects the broader patient populations? So Liz, I'll start with you.
Yeah, so I'll just repeat some of the things I probably already said. I think continued progress is going to depend on improving data completeness and addressing known sources of bias, and creating more consistency in how ECAs are constructed and evaluated, particularly to support the use in the regulatory context. So one of the things I really look forward to about this project is the next step that we will take to write up this experience, and hopefully this can serve as a model for others going forward. And this work can, I think, really be leveraged to help people understand how to make a successful ECA, at least in this context that we considered, which will make it a lot more feasible versus other groups having to potentially start from scratch and developing approaches.
Great. Excellent. Ashita.
I think also when you move beyond regulatory decision making, it's an opportunity to really understand efficacy, safety, and other factors in populations that are broader, to Ava's point, that may not have been eligible for the trial, but in often, we make a leap of faith in those populations when the drugs are on market. And as a provider, like you're thinking about prescribing it to the patient in front of you. So that gives just another tool for us to further our understanding and think critically about each patient in front of us.
Great. Thank you. And Ava, for the patient perspective.
So in addition to making patient data and trial results more reflective of the broader patient community, incorporating real-world data using tools such as external control arms can help reduce patient burden, as well as speed accrual and completion of studies. From a patient perspective, adoption of credible real-world data is important because it connects directly to improvements in patient outcomes and access.
Wonderful. Excellent point. And I think that's exactly why we're all here, is really to improve patient outcomes and access. And the external control arm pilot definitely shows that this is one way we can drive progress forward for patients when it comes to drug development and getting new therapies to them. So we will now shift over to the Q&A. So if you have a burning question that you haven't dropped in the Q&A panel, please feel free to do so. It looks like we have a couple of questions already. So I'll start with the first one. And I'll let my panelists jump in with whoever would like to answer. So first, there is a lot of urgency in rare cancers to accelerate drug development, and external control arms are a promising path. But many patient advocates don't have a clear understanding of how to meaningfully contribute. From your perspective, what are the highest priority actions to facilitate responsible patient data sharing to support the development of external control arms? So who wants to jump in on this one? I see Ava thinking.
I'll jump in because I haven't yet been successful in doing this, but I can tell you what I try to do. One is to really learn about it, to understand it from a patient perspective and understand the barriers that are going to need to be overcome in order to get patients to accrue, and then try to empathize with the barriers to the group I'm working with to why we're not considering doing it. But I think that's really the most important thing, because everything, it's very, very complex. It's very hard to be involved in that part of it for many patient advocates. But the ideas of how attractive in many ways this will be to patients, one, and second, that it's going to happen whether patients really are involved or not, is two. And so I think that becoming partners in this as patients and as patient advocates is really our goal. And that's where I'm trying to be in the conversations.
Wonderful. Thank you, Ava. Ashita, I see you came off mute.
Yeah, I would just add that, I feel like as a community, people who have cancer and their caregivers, they're extraordinary. They're so altruistic when they're already carrying so much on themselves, and they are eager to help and leave behind like knowledge to help prevent the same, hard experience for others. And I think this real world data and just the course of standard care is part of that narrative. It's part of that story. And as much as we can educate and share and create, opportunity for them to learn about this and then, understand the data. And as we move more towards plain language summaries of like studies and things like that, this starts to become really important to engage with advocates, to engage with providers, to engage with patients, and just make sure that this is a known thing.
Absolutely. Those are all great points. And I think just to add to that, in the pancreatic cancer space, I think that the altruism and wanting to give back for future patients so that they don't have to go through the same experiences that patients today are going through is definitely something that we hear consistently from patients with pancreatic cancer, that they want to participate and drive the field forward. So I think there's this opportunity, especially if we are able to use external control arm real world data from patients that are receiving the standard of care outside of a clinical trial and not having necessarily that patient to have to go to the trial site and be able to contribute to research. So I think there's that aspect as well when we're talking about how patients can share and get involved with clinical research when maybe they have some hurdles to actually being able to travel to a trial site. So I think that's a really great point and a really great question. So our next question, what methods are used to adjust for confounding factors? So matching, weighting, stratification. And then how much does the choice of the method matter? And what should patient advocates understand regarding these methods? So that sounds like a stats question, Liz.
I'm gonna say I'm gonna just jump right in and assume that my panelists are also chomping at the bit to answer this question, but I'll jump in first. So we spent a lot of time talking about this in our meetings to define this project, all different ways that we know we needed to address bias and confounding. And so what we landed on was to develop and calculate propensity scores. And there are a couple different ways you can do that, and I won't get too into the weeds on it. But when you do propensity score matching or weighting, what we really did was we defined for each of the patients in the clinical trial control arm their propensity score, which is a one number summary that is really kind of a combination of all of the confounders. We looked at age and ECOG performance status, metastases, number of lines of prior therapy, all those things that go into a patient's prognosis. So we calculated propensity scores for patients in the true control group. And then what we did is we looked for patients in the real world data cohorts to try to match them as best as possible. And there are a couple different ways you can do that. You can do either weighting, you weight by propensity scores, or you can do actual matching by propensity scores. So there's a somewhat rich literature out there that describes this on both the technical and less technical level, but I'll just say we did spend an awful lot of time in approaching this and developing our approach. And interestingly, as I mentioned before, we had some natural sensitivity analyses because we allowed flexibility for different groups to choose different approaches. And what we saw was that some of the groups did propensity score matching, some of them did propensity score weighting, and the number of confounders that were able to be included in the propensity score determination differed because of the availability of data. So there was a fair amount of, heterogeneity happening, which, was very interesting. And so with the results that we've seen thus far, I don't think we can make an answer to say which of those two approaches, the weighting or the matching, would necessarily be better, but a potential next step would be for each of the groups that did matching to try weighting, and those that tried weighting to do matching, and see whether or not the results turn up as consistent as they did in what we've showed you a little bit of already today. And in terms of what should patient advocates understand regarding these methods, I don't know that it's any different from patient advocates than some of my other colleagues in the space, whether it be a medical oncologist or somebody else working on the study team, that there are a lot of statisticians that have developed a lot of these methods. It's very important to have those people at the table and have them involved very early in protocol development to ensure that things like bias are being considered. And just one other thing I'll say about that is, there are a lot of people that are data scientists or data analysts. For a lot of these methods, you really need people involved that are steeped in epidemiology and statistics, not just people who have a lot of familiarity with working with data, because that's where the challenges really lie, and that's where the biases can really be most thoughtfully addressed by people who are trained in the field to understand the implications of not addressing biases. And that could really be devastating in terms of the utility of the results, or the uninterpretability, or really the incorrectness of the results. But we might not know that without appreciating that there was a lot of forethought into how these things were done.
Great. Thank you, Liz. Any of the other panelists want to jump in? Okay, I will move on to the next question, which actually builds on this a little bit. So, Liz, you mentioned a number of the factors that we did the propensity score matching on. One thing we have a question on is, are the data sources reflective of a diverse patient population? So did we see that in the external control arm data sets that we used?
I can start with that. So when we write this up and it's more available, the fuller project is more available. You'll be able to see what we would call table 1, sort of the description of the patients in each of the data sets in terms of things like age and race, ECOG performance status, number of metastatic sites, number of lines of prior therapy, those kinds of things. We did see some diversity in them, and I can't remember all the specifics now, but I know there were some differences by age. There were some differences not just in, for example, the distribution of ECOG performance status, but actually availability of that. So for some of the data sets, we knew that they were a little bit different than others, but for some of the data sets, we just didn't actually know if they were different because of the availability of the data. But I think that in terms of diversity, I mean, there are issues of demographics, but then there are also the issues of the clinical diversity. And I don't know if anybody else remembers, but I believe the trial data patient population was younger on average, but the propensity score approach to matching and weighting does help to alleviate that by selecting patients who it reweights them so that they look more similar.
Another just small point to make is that when you're doing an ECA, depending on the reason why you're using it, this can be a more or less of, what you're looking for. So sometimes we can wait to have the population in the real world cohort look more like the population in the trial. But if the purpose of the ECA is to study the same treatment effect in a population that was excluded from trial, then it becomes harder to disaggregate, like, is this actually a real treatment effect, or is this something else that's confounding that I see? So I think understanding which, tool you dial out, which approach you take, is really going to connect very directly into the reason why you're building the ECA in the first place. And I think that is kind of one of the first principles of the how you would think about approaches to select, and how you would, navigate certain factors within your data. I don't know if Liz wants to add to that, or Ava.
No, I think that's a great point. I think you said it very well. Thank you.
And I'll just add that I think that the big benefit of real world data is that you have the opportunity to really go deep across all of the demographic and disease state patients. And so that, when you, especially looking prospectively, you can set that intent and make it happen.
Excellent points. So the next question, so this pilot was in metastatic pancreatic cancer. So how much can we extrapolate these results to other cancers? Are there differences that we should consider between adult and pediatric populations? So Ashita, do you want to chime in on this one first? You touched on it a little bit in the panel.
I mean, Liz went off mute, so I was kind of hoping
I don't think I ever went on mute was my problem, but I'll let Ashita start, and then I probably have a couple of things I could add.
Yeah, I think with every ECA that you're constructing, with every disease, the way that we approach that disease is different. So if you're talking about something with a very short survival expectation, then OS might be the right endpoint. If you're talking about something where, survival is a much further endpoint, patients are surviving a decade or more, then you might look for something that's a surrogate proxy for OS. And then the complexity of what that surrogate is it imaging based? Is it going to be, based on a more complex formulaic approach to assessing disease, like in myeloma? Is it something else? That will affect how you line things up in the real world data and the trial data. And it's also a bit of a moving target as we evolve the science. So that's kind of one factor. And I think the way that you would assess disease in adults versus children can be different. And the cancers that show up in children are different from the cancers that show up in adults. So there is a lot of applicability from the approaches and how we approach certain aspects of this particular pilot that are bespoke to this pilot, but there are also a lot more lessons and kind of thematic elements to how we approached it that can quite easily lift and inform in other therapeutic areas, and I would argue beyond oncology too. So we are very focused on cancer here, but that is part and parcel of it. And I think this is maybe at the heart of why ECAs aren't just everywhere all at once, is that they are complicated, nuanced, scientifically finicky but important things to figure out. And just acknowledging that plainly here is important. And I thought that was a great question.
Yeah, I like this question a lot too. And I think one of the things that we were able to show with this pilot was that the way that we approached the matching and the weighting based on the baseline factors seemed to do a very good job because, as we've said before, an endpoint like overall survival is very straightforward to measure. And in this patient population, unfortunately, it's a short time to that. So there are not necessarily a lot of intervening things that would necessarily happen to the patients that would vary across being on a trial versus being off trial or vary across centers necessarily. So when I think about, what we can't extrapolate from is to other endpoints. And that I specifically mean because the endpoints that tend to dictate next steps or that are important to patients, or even when they're measured outside of a clinical trial, differ quite a bit from on a clinical trial. So even on a clinical trial, the timing of the scans is very definitively specified in the protocol. And so if you're supposed to get scans every 8 weeks, that means that every 8 weeks, plus or minus, 3 or 4 days, you're going to get a scan. In the real world, a lot of patients would rather delay their scans for various reasons or not. So there's a lot of heterogeneity even in the timing of the measurements. And then beyond the timing of the measurements, when it comes to things that are, where you're looking at benefit or progression based on the tumor burden, what a clinician will say about whether or not a patient is having a response is not the same as what the protocol specified definition of response is. And shout out to Friends' previous pilot. I guess it was a pilot, but maybe it wasn't a pilot, but the real world response project that wrapped up maybe a year or so ago, they looked specifically at comparing RECIST response in clinical trials versus that we would see in an off protocol setting based on clinician reported response. So one of the things that I think we want to lean into are maybe more patient-centered outcomes. We, in a study that we're doing at ASCO, we call it the CDK study, our primary endpoint in that study is not progression-free survival, it is time to treatment discontinuation. And patients can discontinue treatments for various reasons. It could be because the treatment is too toxic and intolerable and they go off study because of that, or go off treatment because of that, or it could be that they have progression of disease. But either way, whether or not you're on a clinical trial or off a clinical trial, the last time you took your CDK4/6 inhibitor is going to have a consistent definition in the real world or on a clinical trial. So I don't think we're really there yet in terms of regulatory approval of things like that, but trying to get to a point where our clinical trial definitions of patient benefit are more consistent to things that we see in the real world, I think really needs to happen for this kind of for ECAs to work well in some of these other settings.
Great. Thank you, Liz. I think those are extremely important points. So the next question, it seems like ECAs can be constructed from many different sources. Are there specific differences that patient advocates should be aware of? And then how do we go from the data that's routinely collected to data that can be used for regulatory decision making? So kind of building on your last point, Liz. So who wants to take a stab at this one?
I could say a little bit about this. I did not contribute, ASCO did not contribute any data to this particular project. And I think most of the data that we used were from large EHR data sources. We have in the past, with collaborations with friends, also looked at data that's been provided by the NCI. And there's also been discussion of looking at claims data. So we did not, as far as I know, do that in this particular pilot, but each of those data sets, those kinds of data sets are going to have potential limitations and differences across them. So that's another important point, I think, in terms of the extrapolation, is that there are different real world data sources that were not investigated in this pilot that could potentially be used, that could lead to different inferences.
I would add to what Liz is saying, and I think maybe it might be helpful to call out that even within the same disease area, there is no perfect data set. And you're going to have to, for any real world evidence project, ECA or otherwise, depending on your question, you probably would go to a few different data partners to assess their data, to conduct a feasibility, to understand patient counts, to try to sift through the variables that matter for the question that you're asking. And you might come up with a different, data set that you should run forward with for in the same disease area for different questions. And I think that's worth acknowledging. And I think for regulators, part of what they're also looking to do, and maybe this is kind of going off on a little bit of a tangent here, is one of the other considerations, because I've heard people say, well, why don't you just take all of the data sets and mash them together? I'm like, well, because there's patients that are anonymized, and I don't know if this patient is showing up in this set and this set and that set, and I've counted them 3 times. So there's all of these different other very practical factors to take into consideration as you're making decisions about what data. And I think from a patient standpoint and from an advocate standpoint, your data is useful. It may not be useful and included in every single type of study that somebody wants to run, but there are studies out there for every chart that exists, I'm sure. And so that's just, I don't know, a little bit of things to think about.
Excellent points. Yes. And I think for just quickly on this, we did have some advocacy organizations, including PanCan, that did provide data as well. And so I think it's thinking through for those advocacy organizations that are starting to collect real world data for their programs, looking at the guidance from the FDA on what meets the regulatory grade requirements so that when you're putting together these data sets, they can be used. So I'll put that plug in for the advocacy organizations out there that are looking at collecting this data. So just 2 more questions. We're getting to the end of our Q&A section. So are there additional roles for ECAs outside supporting settings with high urgent unmet need? So when standard of care remains unchanged, could they be used to accelerate drug development as trials would occur faster? And then to tie into that, are there any characteristics of a clinical trial that could make use of an ECA unsuitable? So I saw Ashita thinking as I was reading.
That's funny. Yeah, I mean, I think that there are probably trials out there where an ECA wouldn't make sense. I can't think of it off the top of my head, but I think there's no absolutes with anything, and I'm sure there's examples of that. I think that there are certain trial designs that are perhaps more amenable than others, but there's just so many different factors to consider. And maybe I'll shift the question a little bit and also kind of something I meant to say on the last one. For regulators too, and I think if there are advocates here who are thinking about building real world data repositories and things like that, I feel like it's important to also mention that FDA wants the data to be auditable. That doesn't mean that necessarily the person who's constructing the ECA at a sponsor needs to be able to see it and track it all the way back to the particular patient, but somebody at some level must be able to disclose that information to the regulators, and that is a big piece of what we're doing here.
Great, thank you. Other thoughts from the panelists on that question? Otherwise, I have Liz, go ahead.
Yeah, I mean, I was just going to say, I know we focus a lot on the regulatory decision making, but in my experience at ASCO, I'm involved with 2 clinical trials that are really much more about trying to look at drugs that are already FDA approved and optimizing them in other ways, making them available for in a precision medicine setting if a drug is approved for patients with a particular genomic alteration, but there are lots of other patients that have a different cancer type. Is it appropriate for those patients? Maybe it's a rare cancer and the drug company doesn't have it's not feasible for them to do trials in this rare population. And then also from a dosing perspective. So I mentioned already our CDK4/6 inhibitor study. There are a lot of uses and interest in understanding, optimizing care from the perspective of what's the best way for patients to receive drugs that are already approved. For example, in patients who are older or have been identified as vulnerable with multiple comorbidities or frailty, should they really be getting the full FDA approved dose of these drugs? So I think that there are lots of different ways that we can think about ECAs, not just from the drug. When I think about drug development, I think about pre-approval, but also in the post-approval setting, because there's lots of real world data about how these drugs are actually given and used, and sometimes they aren't consistent necessarily with the intended use. And that's where we get a really great, real world experience with them. There are a lot of comparative effectiveness questions, I think, that could be addressed using ECAs as well.
Great.
Little off topic maybe, but I don't know. I think about it a lot because of the studies that we have at ASCO.
Thanks, Liz.
There's
Ashita, did
also
you have something to add?
Yeah, I mean, I think Liz brought up something really important, and I think it's worth mentioning. One of the celebrated examples, is not a J&J product or drug at all, but like the Ibrance example where the drug was approved in male breast cancer, not because they did a trial in male breast cancer patients, it's because they had enough men in the real world who took this product for an indication where there was nothing for them. And on the basis of that, they were able to review the information, see that it was robust, and I think they were able to expand their label. Like that's a very high bar to meet, but there is examples of success that go to what Liz said. That was an example of a product that was available on market and approved, right?
That's an amazing example. Thank you for bringing that up. So one final question as we end our Q&A, and I'll go clockwise on my screen. So what do you hope patient advocates take away from this webinar? So Liz, I'll start with you, and then we'll go to Ashita and then Ava.
I guess there's a really great outlook for using this approach and this technology. And like with many things, I get so humbled when there are many people from many different expertise, backgrounds, companies, stakeholder groups that are all willing to come together and give a piece of themselves, a piece of their organization to really further this. And so, it really takes a village. And I would say that Friends does an amazing job of bringing people together to work together, people who may not otherwise have the opportunity or necessarily the motivation to do it. So I think the future is bright for ECAs in cancer.
Thanks, Liz. Ishita?
I would echo what Liz said. I think this is just a really great project, and I would say we need partnership with advocates and with patients, and that, learning more about these sorts of tools and spending more time with statisticians is always a good thing.
Excellent. Thank you. And Ava?
I agree with what everyone else is saying. And the one other piece is that then, especially as we go into prospective uses, to look for ways that patients can be partners in the research and bringing in PROs and other things where the patient voice is a part of it as well.
Excellent points. Thank you all to the panelists. This was a great discussion. Honored to moderate this. And thank you for Friends for bringing all of us together, not only for this pilot project, but also for this webinar. So with that, I will hand it back to Bernat.
Awesome. Well, thank you all so much. Really appreciate all the panelists and Cassidy for moderating. I would just encourage everyone to look at our ECA website, where we'll continue to update as we continue moving forward with our pilot project. I know that we had some questions about when these results are going to be published, so stay tuned for that. And please check our events page as well. We have a couple of upcoming events in May and in June on unlocking the next generation of therapy. So we hope to see you there. And thank you all so much for joining us today.